And just to remind everybody that we will, if you don't want to be recorded, we'll have it on speaker view for the recording. So you feel free to turn off your video, rename yourself, whatever other things you want to do.
Yeah. Okay, right, so let's get started. So this is session 5, which will be a continuation of proteins. So we're going to be talking a little more about protein folding and post-translational modifications. So what happens after translation and how proteins are controlled by other factors. And then for part 2, we're going to go into some more protein-related techniques. So we'll be talking about co-immunoprecipitation, immunofluorescence, fluorescent tagging, which are all involved in looking at protein-protein interactions as well as protein localization. And then we're going to go into mass spec, which is a way of identifying proteins and basically sequencing amino acid sequences. And then we're going to go into techniques for determining structures of proteins. So we'll be talking about X-ray crystallography and cryo-electron microscopy. Okay, so going to go to protein regulation first. So last week we were talking a bit about protein structure. So there are at least 3 levels of protein structure. Sometimes there's 4, depending on which protein you're talking about. So the primary structure is just the amino acid chain sequence. This is just a series of amino acids that are attached by peptide bonds. These amino acids have side chains that can undergo hydrogen bonding amongst each other, and these hydrogen bonds can form secondary structures, either beta pleated sheets or alpha helices. And then once the secondary structure is folded, the cysteine residues, which contain a sulfhydryl group, can form disulfide bridges, which are represented here. And this comprises the tertiary structure. And then certain proteins exist in complexes, and these complexes are called quaternary structures. So these are a combination of multiple tertiary structures. So the process of protein folding starts during translation, as the amino acid polypeptide is exiting the tRNA ribosome complex. The folding process starts, and because this takes place in the cytoplasm, usually how this works is the hydrophobic residues will gravitate towards the core region of the protein, while the polar side chains will be on the outside. However, during this process, intermediary folded protein forms can be created, and these can, depending on what protein it is, these can form aggregates, which is something that you don't want. This is similar to what we have in prion diseases like mad cow disease. In addition, misfolding can occur as well, and sometimes when there's a long series of hydrophobic side chains, this isn't energetically favorable in aqueous environment like the cytosol. So in order to work around these challenges, there are proteins referred to as chaperone proteins that are involved in assisting the protein folding process. So these are evolutionarily conserved. So they're present in eukaryotes and prokaryotes. So the ones that are in parentheses here, those are the eukaryotic equivalent of the prokaryotic chaperone. There are multiple forms, and you'll notice that a lot of the names, they start with HSP. That stands for heat shock protein. The reason why it's called that is because these proteins are generally they're generated at higher rates following a heat shock event. And the reason for this is that exposure to high heat can result in protein unfolding or misfolding. So the cell compensates for this by producing more heat shock proteins, which can then assist in refolding of the proteins to a more stable form. So there's some major examples of heat shock proteins. I'm only going to focus on two types here: HSP60, which in prokaryotes is called the GRO-EL complex, and HSP70. The rest are pretty similar, so I'm not going to go into too much detail with the rest of them. So, so these two types of chaperone proteins have different mechanisms. So the GRO-EL, GRO-ES system, which is the equivalent of the eukaryotic HSP60 complex, is in the form of this barrel-like structure. So so this barrel essentially acts as an isolation chamber, which prevents misfolded proteins from forming aggregates. So the misfolded protein enters through this opening in one end of the barrel. Usually only one side of this complex is in use at any given point. And then this cap-like structure is added, and then an ATP hydrolysis takes place, and this is and the energy produced from this hydrolysis reaction is what causes the protein to be refolded in the correct manner. And then in step 3 here, this better folded protein is now released, and on the other side of this complex, a new improperly folded protein can enter, and the cycle can repeat. In contrast, the HSP70 chaperone system starts acting as translation is occurring. So so after about 7 hydrophobic amino acids are produced during the translation process, the HSP70 complex binds to the growing polypeptide, and this occurs as the polypeptide chain is leaving the ribosome. Similar to the HSP60 complex, this system uses ATP hydrolysis to promote proper folding of the polypeptide. So in this case, the molecule that's designated as the client is the polypeptide, and the mature client is the properly folded peptide. Any questions about chaperone proteins?
Yeah, I put it in the chat. The first example you gave, how does it, so you show the misfolded protein that's in there, but how does it recognize that it's misfolded, and as opposed to just it's correctly folded and then it unfolds it?
So it depends on what the problem is. Sometimes they form aggregates, in which case it could detect if there's aggregate formation. As far as just it being energetically unfavorable, I'm actually not sure, so I can look into that.
There's also some sort of signaling pathway, and I'm just like so foggy on the details, Shirley, so I apologize that I'm like bringing this up without actually telling you. But something about the unfolded protein response and like ER stress. So there can be like a lot of proteins misfolding. There's actually like signaling that gets activated when the ER is like overwhelmed. But I can't remember any more details than that.
The ER is the endoplasmic.
Yeah.
Which is on where is the endoplasmic reticulum? What is it? Reticulum?
Reticulum. So it's on the edge of the nucleus.
Oh, okay.
Yeah, so it shares a membrane with it.
Oh.
Yeah, I'll see what I can find on that and put it on Google Classroom once I do.
Great, thanks.
Yeah. Okay, so I'm going to talk about post-translational modifications now. So this is a step that occurs after a protein has been fully translated and folded. So as you can see, there are a number of possible modifications that can take place after a protein is translated. So this can include acetylation, which is the addition of an acetyl group, phosphorylation, which is the addition of a phosphate group, methylation, ubiquitination, disulfide bonds, which we kind of talked about in the context of bonds between cysteine residues. I'm not going to talk about glycosylation, hydroxylation, lipidation, or simylation, just because there's too much content to cover. I'm going to focus on these four: acetylation, phosphorylation, methylation, and ubiquitination, just because they have probably the more important consequences as far as gene expression and protein regulation is concerned. So acetylation and methylation both have are both involved in gene expression in the specifically when we're talking about modifications of histone proteins. So you may remember from session 2, histones are proteins that form complexes with DNA to form nucleosomes, and this is how DNA is stabilized and packaged in the nucleus. You also may remember that histones are they're rich in lysine and arginine residues, which are targets for acetylation and methylation modifications. So we're going to talk about phosphorylation. So phosphorylation can have multiple effects on protein function. In the context of proteins that are enzymes, phosphorylation can cause conformational changes that can alter the function of the protein or its ability to function at all. It can also affect stability, which is something I'm going to talk about in more detail soon. In addition, I'm going to be talking about ubiquitination, which is the addition of a small molecule called an ubiquitin molecule, and this controls protein stability. Before I get into detail, I just also want to go over which enzymes or molecules are responsible for these modifications and where which amino acid residues are being targeted here. So acetylation is carried out by enzymes called acetyltransferases, and methylation is carried out by methyltransferases. Both of these specifically target lysine and arginine residues, which you may remember are both positively charged residues. Phosphorylation is carried out by kinases, and the residues that are targeted are serine, threonine, and tyrosine residues. So serine and threonine are both polar uncharged amino acids. Tyrosine is a hydrophobic amino acid. So there are multiple types of kinases. Many of them are capable of phosphorylating both serine or threonine residues, but often tyrosine phosphorylation is carried out by kinases that specifically target tyrosines. In addition, the tyrosine phosphorylation is also associated a lot of times with intracellular signaling, because this is something that often occurs at the membrane. Tyrosine being a hydrophobic residue, it's often found at membrane receptors. And the last modification I want to talk about is ubiquitination, which is carried out by ubiquitin ligases, and this specifically targets lysine residues. Acetylation and methylation are two modifications that can occur at the histone proteins. So again, acetylation is just the addition of an acetyl group, and methylation is the addition of a methyl group. So acetylation is carried out by histone is carried out by HAT enzyme, which is a histone acetyltransferase, and they add the acetyl group from acetyl CoA molecule. And what this does is it causes the chromatin to essentially open up, and this allows access to the chromatin by RNA polymerase, and this promotes transcription of a gene. In contrast, methylation, which is carried out by methyltransferase, in this case, it's the enzyme is referred to as PRC2, adds a methyl group to the histone, and this causes the chromatin to condense, and this prevents access to the chromatin by RNA polymerase. So this essentially turns transcription off. Next, I'm going to talk about deubiquitination, which is one of the mechanisms for promoting protein degradation. So, ubiquitination is carried out by 3 enzymes that work in conjunction with each other. They're referred to as E1, E2, and E3. So E1 is an ubiquitin activating enzyme. Ubiquitin is a small molecule that consists of 76 amino acids that has a lysine-rich C terminus. So this ubiquitin is added to, it binds to the E1 enzyme in an ATP-dependent manner. So this reaction uses energy in the form of ATP. The E1 enzyme then transfers the ubiquitin molecule to E2, which is the ubiquitin conjugating enzyme. E2 then forms a complex with E3, which together form the ubiquitin ligase complex. The E3 molecule then recognizes proteins that are marked for degradation by detecting signals or motifs, which I'll get into a little more detail in a bit. But what happens next is that E3 then helps E2 form a polyubiquitin chain and also connected to the target protein at a lysine residue. So how this works is the C-terminal end of the ubiquitin molecule is bound to a lysine residue on the substrate protein. The next ubiquitin molecule is then attached to a lysine residue on the previous ubiquitin molecule. And by doing this, you end up with a protein that's marked for degradation by a polyubiquitin chain. This polyubiquitinated protein is then detected by a complex called the proteasome. So the proteasome contains ATP-dependent proteases. It is a complex that consists of a hollow cylinder, and inside the cylinder are protease molecules, which are represented by these red dots. So the active site of these proteases are facing the interior of the cylinder. At either end, there's a cap, and each cap contains a 6 subunit ring domain that the protein that's targeted for degradation is threaded through. And then in an ATP-dependent manner, the protein is then degraded by the proteases inside the complex. And there are a number of ways of controlling the ubiquitination process. So one is phosphorylation of the E3 enzyme, which activates it. E3 is also subject to conformational changes by binding other molecules, which can reveal or obstruct the active site of E3. In addition, the E2, E3 complex detects recognition sequences on proteins. These are sometimes called destruction box domains, or there are also equivalent motifs called Ken boxes. There are multiple motifs involved in this process, but essentially these proteins have amino acid sequences that mark it for recognition by an ubiquitin ligase. And another mechanism for recognition of a target protein by ubiquitin ligase is called the degron, which are formed by phosphorylation events. So phosphorylation is the addition of a phosphate group by a kinase. And this can be used as a mechanism to control protein degradation. So one mechanism for this is the creation of phosphodigrons. So here kinases add add phosphate groups to a protein sequence in a pattern that allows for the ubiquitin ligase to recognize this phosphorylation pattern, and this promotes recognition by ubiquitin ligase complex, resulting in ubiquitination of this protein, resulting in its degradation. Conversely, phosphorylation can also prevent protein degradation. So as I mentioned before, there are motifs that in amino acid sequences that ubiquitin ligases can detect. One of these types of motifs is called a destruction box or a D box. So the addition of a phosphate group to a D box can prevent detection by ubiquitin ligase, thus stabilizing the protein. This phosphorylation event can be reversed by another type of enzyme called the phosphatase. So phosphatase removes a phosphate group from a target protein. This would remove the phosphate group that's blocking the destruction box, which would promote ubiquitination, resulting in degradation of the protein. So phosphorylation, depending on where it's located, can either promote or prevent protein degradation by an ubiquitin ligase complex. So any questions about protein degradation or any of the modifications we just talked about?
So we're actually, the system is trying to degrade on purpose those proteins, is that right?
Yeah.
Very. All right.
Okay, so we're going to talk a little bit about techniques now. So I'm going to start with a technique that examines interactions between two proteins, or protein-protein interactions, and this is called co-immunoprecipitation. So the basic concept here is that you have proteins that exist in a complex, and you then basically pull down or immunoprecipitate out one protein of interest. And you do this using beads that are usually conjugated to an antibody or some other molecule that has affinity for the protein, the first protein of interest. One way of doing this is to include a tag or a DNA sequence at the end of one of the proteins of interest, and this will basically add on an antigen that is recognized by commercial antibodies. So you have these beads that are conjugated to an antibody, and this will bind to one of the two proteins that you're interested in. After a series of wash steps, you essentially purify that first protein of interest, and it will still be bound to whichever other proteins it was bound to in the cell. So then after a series of wash steps, you elute the protein from the beads, or sometimes it can be, this step isn't totally necessary. And then you analyze using SDS page, gel, or Western blotting. If this is successful, what you should see is your protein of interest that you immunoprecipitated, along with any other proteins that it was bound to. One way of confirming this is to also tag a second protein of interest that you are predicting is in a physical interaction with the first protein. So this is an example of one that works successfully. So these were two proteins that were examined in this figure. So one is called ESP1, and it was tagged with an antigen called a MIG tag. And beads that were conjugated to an anti-MIG antibody were used in this experiment. And a second protein, which is referred to as PDS1 here, was tagged with another antigen called HA. So basically what was done here is that these cells that were of different strains were broken up, they were lysed, and the cell lysate was it was used in and in this type of experiment where it was incubated with beads that were conjugated to with an anti-MYC antibody. After several wash steps, this one protein that was tagged with MYC was isolated along with any other proteins that it was bound to. And then the sample was then run on SDS page gel and blotted for both using antibodies both for MYC and for HA. So if there is a physical interaction between the two proteins, there should be a signal coming from both from members that were probed with both the MYC antibody as well as the HA antibody, which is what you see in these two lanes here. So the actual question that I was trying to answer with this experiment was if the affinity between these two proteins has changed when this CDC55 gene was mutated. So you can see here that the amount of PDS1 that came down with ESP1 in this pull down, it increases when this was mutated. So that was my big takeaway from this experiment. So what you're seeing here is these first three lanes, those are just the cell lysate before any treatment with any antibody conjugated beads. So you can see there's the proteins are present, but they're not, but you don't see as strong a signal. The reason for that is this is essentially a concentrated amount of the protein that's being pulled down by the beads. So yeah, the short version is you pull down one protein using antibodies, and you then run it on a gel and check for whichever other proteins are pulled down along with it, and that confirms a protein-protein interaction. Does that make sense, or any questions about that? Okay, another way of examining multiple proteins at once is called immunofluorescence. So this gives some insight into whether or not two proteins are interacting, because what it does is it shows you where in the physical cell or tissue a protein is localized. So if you have two proteins that are localized in the same vicinity, or they're co-localized, that may indicate that there's a physical interaction, but it doesn't definitively state so. But this is useful in getting some sense of whether or not there's a physical interaction, and it also is a really useful way of seeing where in the cell a protein is localized. So, so in immunofluorescence, cells or tissues are they're fixed, which means that they're treated with a cross-linking agent, which basically maintains the structural integrity of the cell, so that everything is in the same location as it was while the cell was alive. And the membranes are permeabilized to allow antibodies that are conjugated to fluorophores into the cell. So an antibody that's specific for a specific antigen enters the cell, and it targets the protein of interest or the antigen of interest. There are two ways of doing this: primary and secondary immunofluorescence. This is similar to what we were talking about last week with Western blotting. Primary immunofluorescence basically just means that the primary antibody is conjugated to a fluorophore, which can then be visualized using fluorescence microscopy. Secondary immunofluorescence just means that you have a primary antibody that's not conjugated to anything that can be visualized, but instead you have a secondary antibody that detects the primary antibody, and the secondary antibody is conjugated to fluorophore, which is visualized under fluorescent microscopy. And one thing that can be done is two different proteins can be detected by immunofluorescence at the same time to see if there's a potential interaction between them. So one of the downsides to immunofluorescence is that you're working with cells that have been fixed, so they're not living cells. So there's one way around this is to use in vivo fluorescent tagging. So what's done here is sequence for a fluorescent protein is added to the sequence for your gene of interest. This gives you a fusion protein, which consists of your protein of interest along with a fluorescent protein. A really commonly used one for this is GFP, or green fluorescent ablated, from jellyfish. There are variations of GFP that appear as different colors by fluorescent microscopy. So there's also red fluorescent protein, there's cyan fluorescent protein. So again, multiple genes can be tagged with different colors of fluorescent proteins, and this may give you some insight into whether or not there's the protein-protein interaction or if they're co-localized. And this is an example of how this would appear under a fluorescent microscope. So here, the protein that was tagged with GFP is called TUB1. This is tubulin. This is what makes up the spindle that separates the two copies of DNA during mitosis. And these were two different strains of yeast cells that these were used in, and you can see that using fluorescence microscopy, you can visually see where the spindles are. So that's what these green dots and lines represent. Okay, so I'm going to go into mass spec, which is a way of identifying unknown proteins, as well as a way of determining amino acid sequence and to identify post-translational modifications. One method of mass spectrometry is called MALDI-TOF, and so here what takes place is proteins are isolated usually from some form of electrophoresis. The example shown here is two-dimensional electrophoresis, where the proteins are separated by first by size and then by isoelectric point, essentially by electrical charge, and a single protein spot is removed from the gel. This is also sometimes done in one-dimensional electrophoresis as well, where they're just separated by size. This protein is then removed from the gel and broken down into short peptides and mixed with an organic acid and dried. A laser then ejects the sample in the form of an ionized gas, and each peptide molecule is then carrying one or more positive charges. These ionized peptides are then accelerated in an electrical field, and they enter a detector. And the speed at which the peptide reaches the detector is dependent on both its mass and its charge. So the larger peptides will move more slowly, and the more highly charged peptides will move more quickly. And the peptides that contain a single charge, the mass of these peptides can be accurately measured. So the masses of all the peptides are then collected, and they're used to compare against a genomic database. So these databases contain information on the masses of all proteins present in an organism and their predicted peptide fragments. And using these databases, the protein in question can be identified. So that's how mass spec is used to identify unknown proteins. They can also be used to sequence an amino acid chain and to identify post-translational modifications. And this is done using a technique called tandem mass spectrometry. And this uses two mass spectrometers to determine the amino acid sequence of individual peptides. So here a single protein peak is taken from column chromatography, and then high energy gas atoms are used to fragment the peptides even further. And this leaves peptide fragments in the form of a ladder, where each peptide fragment differs by a single amino acid. And the second mass spectrometer then separates these smaller fragments and detects their masses. So the difference in mass between these fragments is is the mass of a single amino acid. So these masses can be used to deduce the amino acid sequence in this way. In addition, the changes in mass that are associated with different post-translational modifications are also known, and these can be used to determine which sites have modifications on them. So that's basic multi-top and tandem mass spec. Are there any questions about that?
Yeah, surely the first system, the MALDI-TOF, you don't actually get the sequence from that, right? It's only by then comparing it with the databases.
Right.
Okay. If you actually need to know the sequence, if it's like a protein that's not in the database, then you have to use tandem mass spec.
Yeah.
Okay.
And is tandem more expensive? Is that why we use it less often, or is it really just dependent on the question that you're asking?
I think it depends on the question.
Yeah. That makes sense.
Okay, so gonna go into protein structure now. So I'm going to talk about two techniques for determining protein structure. I'm going to talk about X-ray crystallography first, and then cryo-electron microscopy. So the majority of the protein structure information that we have right now comes from X-ray crystallography work, but a growing amount of protein structure knowledge is currently coming from cryo-EM work. I'm going to talk about X-ray crystallography first. So how this works is a narrow beam of parallel X-rays is directed at a sample, which is a crystallized form of the protein, which is created by purifying protein of interest in large quantities, and then performing a protocol to form a crystal structure. This beam of X-rays is directed at the protein crystal, and most of these X-rays will pass through the crystal, but a small amount of it will be scattered. This yields an X-ray diffraction pattern, and this X-ray diffraction pattern will give the position and intensity of various spots, which will give information about the location of atoms within the crystal. The analysis of this diffraction pattern will then yield a 3D electron density map, which can be used to deduce the protein structure. Some of the major disadvantages of X-ray crystallography is that it requires an organized protein crystal, which requires large quantities of ultra-pure protein. This doesn't work as well with larger or less soluble proteins, for example, membrane proteins. And it only gives a representation of the static form of a protein as opposed to a dynamic one. One way around this is, around these drawbacks, is the use of cryo-electron microscopy. So cryo-EM starts out the same way. It involves sample purification, and then the purified sample undergoes negative staining. So negative staining involves placing the sample on a thin carbon filter and then mixing it with a heavy metal salt. As a result of this, the salt will cover the carbon film everywhere except for where the sample is present. So this yields a negative stain. So negative stain electron microscopy is limited by resolution of the smallest molecules present. So what cryo-EM does, it allows direct visualization of these molecules. This sample is then suspended onto an electron microscope grid, and and it forms a thin film on the grid. The specimen is then rapidly frozen using a coolant, and the hydrated cryo-EM then allows for visualization of the molecule itself rather than a negative stain. Multiple images are then obtained, and then these images are combined, and they are averaged to reduce the background noise. This process is called single particle reconstruction, and this gives a 3D model of a single particle. So some of the advantages of cryo-EM over X-ray crystallography is that it requires less sample and purity isn't as important. The protein doesn't need to be crystallized, and this rapid freeze protocol allows for the protein to be analyzed while it's still in a near-native state, as opposed to X-ray crystallography where it's in a static form. The major disadvantage to cryo-EM is that it has lower resolution than X-ray crystallography. Any questions about structural techniques?
So, so what is the resolution of cryo-EM? I mean, X-ray crystallography, you get down to the individual atom positions, right?
Yeah.
And what about cryo-EM?
I think it's about 0.3 nanometers in general, but for a single particle, it's around 0.5 nanometers, if I'm remembering it correctly.
So what does it mean in terms of figuring out the protein's structure? Like what do you actually not, what can you not see? Is this the positions of the atoms are not that clear, or you can't get down to the individual atom, you just see like clumps of them, or?
The impression I get is that the main issue is background noise. It can sometimes obscure some of the outer details of a protein structure.
So we're trying to get some sort of model, correct? Unfolded model of the protein.
Yeah.
At the end of this. Okay, cool. Do you always get a model, like an unfolded model, with this process? Or depends on the computer?
Yeah, it depends on the sample as well, as well as the protein. There are some proteins that this still just won't work for, because in either technique you do need pure protein in larger quantities than most techniques would require. So there are always some challenges when it comes to obtaining purified proteins. Yeah, and depending on the structure, certain proteins, neither technique will work for. If a protein has large stretches of disordered regions, that can definitely be a problem for both cryo-EM and for X-ray crystallography.
How do we ensure the purity of the protein, of the sample protein?
And the best way to confirm, I think, is by mass spec. If there's any other proteins that are present in your sample, mass spec data will show that. Yes. A cheaper way of ensuring purity is to just take your sample and run it on a gel. If And then when you stain your SDS page shell, you should only see one band corresponding to the protein that you're trying to purify. If you see additional bands, then that means you have other proteins in there. Depending on the quantity of protein, that you may or may not actually see that, because could still be present in small quantities. So really the best way of confirming that your sample is totally pure is by mass spec.